The overall goal of this procedure is to prepare recombinant protein from non-pathogenic bacteria to facilitate biophysical chemical studies, such as a protein X-ray crystallography or enzymatic kinetics.
This method can help answer key questions in how to prepare recombinant protein of interest using non-pathogenic bacteria for preliminary biophysical characterization. The main advantage of this technique is that biophysical characterization for protein of interest can be done in any laboratory equipped with common instruments, such as cell culture incubators and spectrophotometers.
To begin this procedure, prepare the ligation reaction as outlined in the text protocol. Pipette 3 microliters of the prepared ligation reaction into a tube containing 50 microliters of chemically competent E. coli DH5 alpha cells. Using a pipette, gently mix the solution. Ice the solution for 30 minutes. Next, heat shock for 45 seconds at 42 degrees Celsius. After this, add 1 milliliter of LB medium and grow the cells while shaking vigorously at 250 rotations per minute at 37 degrees Celsius for 45 minutes. Spread 100 microliters of transformed cells onto an LB agar plate, supplemented with 100 micrograms per milliliter kanamycin. Incubate for 18 hours at 37 degrees Celsius. Using a sterile tip, pick colonies from the plate. Grow the cells in 3 ml.